Hi, my name is Talia Hardin. Nirenberg and Mathai were the first scientists to determine that a specific RNA sequence coded for a specific amino acid back in 1961. They determined this by performing an experiment. The experiment consisted of a cell-free system so that translation can happen outside of the cell. They did this by adding a homopolymer, in this case poly-U, to a test tube in which one was labeled as radioactive and the other 19 were unlabeled amino acids. The translation took place and the protein was filtered to check for radioactivity. After that, the experiment was repeated 20 times with each tube and their corresponding amino acids. Their key findings from this experiment is that they were able to figure out that UUU was the specified amino acid that was newly synthesized by the homopolymer. With that, they were able to determine that UUU encodes for phenylalanine, all in which plays a role in the structure of DNA. They were also, by using that experiment, they were able to figure out that AAA encodes for lysine and CCC encodes for proline. Nirenberg and Leder continued experimenting to crack the genetic code by forming a separate hypothesis from the first one that was discussed. The scientists wanted to know what other matches between codon and amino acid could be determined based off of the use of tRNAs. They figured this out by using an already known codon, synthesized it, and added it to a mixture of ribosomes and the tRNAs attached to the amino acids. The mixture was then passed through a filter to determine which amino acid is attached to the mRNA and tRNAs. They came to the conclusion that the genetic code can be deciphered because mRNA with GUU was used and filtered and was bound to valine. Therefore, that codon specifies for GUU. The experiment was carried on to determine the other mRNA codes. These findings were very important for gene coding, DNA structure, gene expression, translation, and etc. Thank you very much for watching.